Let's go ahead and get started. I'm Carrie Bourne. I'm from the Office of Continuing Education at UW-Whitewater, and we host the Fairhaven Lecture Series here at Fairhaven Senior Services and have been doing that since 1983. We've held hundreds and hundreds of lectures for thousands and thousands of people, and we're thrilled to be here again today. This semester, our theme is Science and Society. We're hosting lectures by our science faculty from across the Whitewater and Rock County campuses. And today we have another one of our physics professors. Dr. Martin Tchernookov received a bachelor's degree in physics from Caltech and a doctoral degree from the University of Chicago. He has held postdoctoral positions at Emory University in Atlanta and McGill University in Montreal. His research interests lie in the field of computational statistical physics, particularly in regard to problems of biological relevance. He is also a proponent of the open source software community, manages a cluster of Linux workstations, and develops apps in his spare time. He works with students on digital electronics and coding projects. Please welcome Dr. Tchernookov. Tchernookov, I'm sorry, thank you.
Thank you for having me here today. I have a bit of an accent and a bit of a cold on top of it, so if you don't understand what I'm saying, please feel free to interrupt me and just ask. Sometimes my students get annoyed with me, especially when I have a cold. Anyways, so today I want to talk to you a little bit about what I do and what my PhD was about, and a field which has become really relevant in the last 30 or 40 years. It's a very interesting interdisciplinary field which overlaps different sciences, different fundamental sciences into one very interesting subject that tries to resolve a lot of... How is it now? Good. So biophysics is an interdisciplinary field which overlaps a lot of sciences: chemistry, biology, obviously, physics, and mathematics. And it tries to solve a lot of problems that exist in regards to living organisms, the human body in particular. I do not claim to be a biologist, so I'll be saying some biology things, and very likely some of them might not be 100% true. Please don't hold me up to that. But I want to talk to you more about the history of the field and a little bit more about what I do on the physics side of the field. All right, let's get started. So here's an outline of the talk. I'll talk, as I said, a little bit about the history of biophysics as a subject, the relationship between biology and physics, some of the differences between biology and physics. I'll talk about some of the famous problems that people have been trying to solve for a long time in biophysics. I'll then talk more about the statistical physics side of things, information processing in particular, and then if there's time, I'll talk about some of the actual work that I do here at UW. All right, I like to start my talks, especially the ones that are not too technical, I like to start them with this quote from one of my favorite sci-fi authors, Isaac Asimov. There's actually, so this is a quote from a series of books, probably the books that made him really famous, the Foundation series. And I like these books. I like Isaac Asimov. He was a scientist himself. And even though he wrote over 70 years ago, a lot of his stuff is still relevant today. And actually Apple TV is making a series about the Foundation books that is hopefully good. So what is the quote about? Thus it was established thousands of years ago that the universe as a whole, in its full complexity, cannot be represented by any simulation smaller than itself. In other words, if we want to model the universe on a computer by making guesses about how it works, writing those guesses down on a piece of paper and then coding those guesses, those equations, into a computer, the size of the computer that we're going to need to perfectly predict nature and the universe and all its laws is going to be as big, if not even bigger, than the universe itself. That's one of the biggest concerns that we have as physicists. Are we overstretching ourselves too much. And that is why I like this quote so much, because it didn't come from a physics textbook, it came from a sci-fi novel. Yet it really encapsulates the fundamental philosophical struggles of any physicist who is trying to solve any deep problems that we as humans have discovered. I'll start a little bit with the historical context about biophysics, the history of biophysics. And I'll start by saying that physics has been lending tools to other sciences to use for a long time. I have in front of you two pictures. On the left, I have an artist's representation of the DNA structure, the helical structure of the DNA molecule, something which biologists have known for 80 years now. And on the right, there is the image that was published of the X-ray crystallography experiment that was performed, a physics experiment that was performed on DNA molecules in order to discover or discern the structure of the DNA molecule. So physics lended a tool to a biologist, to a couple of biologists actually, that was used to discover the structure of a biological molecule, one of the, arguably the most important biological molecule, the DNA in our cells. But the interactions between physics and biology in terms of tools that physics provides to biology are not just limited to the DNA molecule and X-ray crystallography. By the way, just a little bit about how that image on the right is taken. How many of you have had an X-ray in their lives? I think all of us at one point or another we have had an X-ray. And so the way this image is taken is very similar to the way we as humans go to the doctor and get an X-ray taken. We have an X-ray machine, a very powerful machine in this case, which shines X-ray light onto a target. The target in this case being a sample of DNA. And on the back of the target there is a film, there is a photographic film which is very sensitive to X-rays. And that's the image that gets formed. Notice the white circles with the black stripes in the image, the cross that appears on the right-hand side. That's a result of the scattering of the X-ray light off of the DNA molecule. As the light passes through the DNA molecule, it scatters in this pattern and it gets projected onto the film behind. And then the brilliancy of the people who discovered the DNA, particularly the lab technician who worked on this, was to figure out that, oh, the three-dimensional structure that gave rise to that image on the right came from a helical molecule. And that's how the DNA was discovered. Again, there are other tools that physics has provided to fields like biology. I have on the right an image of one of the earlier production models of electron microscopes. Electron microscopes are used to measure image very, very small things, including living things like a virus. On the left, we have a real, although computer-enhanced, but still real image of a virus. Can you guess what kind of virus we have on the left? It's the coronavirus, but typically in the news you see it in this artist representation with the spikes really clearly pronounced. So historically, biology and physics have had a lot of overlap. Mostly, well, at least in the early years of biology, mostly through providing tools that can be used in observing and measuring living systems. But physicists have been interested in the universe, including the living organisms in the universe, for a very long time. And so I include this quote in my talks about, that came from a book written by one of the founding fathers of quantum mechanics, the basis of all modern physics and all modern understanding of the universe that we have as physicists. Erwin Schrödinger, who gave us the Schrödinger equation in quantum mechanics, wrote in his book, What is Life, he conjectured that there might be some crystals that exist in a living system, in a living cell, that encode information about the living cell. Here's the actual quote, the actual question that he posed in his book. How can the events in space and time, which take place within the spatial boundary of a living organism, be accounted for by physics and chemistry? He wanted to ask, how can I explain life knowing what I know as a physicist or as a chemist? And indeed, so the quote that I showed you in the book was from 1944, and in 1953, around the time the DNA structure was discovered, one of the people who discovered the structure, Crick, he actually sent a letter. Well, there weren't emails at the time, so he sent a paper letter to Schrödinger thanking him about the book, that how it motivated him and how it excited him and made him want to go and pursue biology, and how his prediction of the existence of these special crystals inside a living cell, how that prediction is actually coming true through his own experiments. Indeed, if you look at some of the major Nobel Prizes, the major milestones that have been awarded Nobel Prizes in biology, in physiology, the discovery of the structure of the DNA by James Watson and Francis Crick, the neuron mechanism, meaning the action potential, which is what allows our neurons to work with each other, discovered by Alan Hodgkin and Andrew Huxley, and the evolutionary mechanism of passing gene information from one generation to the next, discovered by Salvador Luria and Max Delbruck. These 6 individuals who got Nobel Prizes for biological discoveries, 3 of them actually were either a physicist or a wannabe physicist. Crick, I believe, was actually a physicist by training. Huxley wanted to be an engineer, and Delbruck actually got a PhD in theoretical physics, yet they got interested in biology and then went on to do work in biology and got Nobel Prizes by discovering really fundamental results about how living systems work. I want to move on to some of the problems that are currently discussed in biophysics, and the problems that we as scientists believe physics and other fields, mathematics and chemistry, can really help biologists in their attempt to answer them. Some of the more, I looked actually, while I was preparing this talk, I looked around on the internet. I wanted to see what are the 10 most relevant and most interesting problems in biophysics. And it turns out that no one agrees on that. Everyone believes, that what they're working on is more important than what the next person is working on. But I sort of synthesized a short list of 10 questions, 10 problems which I think are important. Gene regulation, how genes activate and inhibit each other and help propagate information from one generation to the next. Protein dynamics and protein function. We are all made up of, a lot of our bodies are made up of protein, and those proteins are very important because they perform a lot of functions in our body. They move things around, they activate things, they close things off. They have a really important meaning in our bodies, but we don't know why and how, and sometimes we can study a protein and not know what its purpose is, so people try to answer such questions. Membrane transduction of signals. Cells communicate with each other, not the way we do at the higher level. At the higher level, we function. Cells don't write letters to each other, they don't talk to each other, but they communicate through chemicals, through biochemicals. And that's a very interesting question, why they do it, how they do it. Are they optimal in their communication, or are they wasting a lot of energy talking to each other? These are all questions that people who study membrane transduction are interested in. Biomechanics, why our bodies are the way they are. Why do fish have fins? Why do we have legs? How is our skeletal structure relevant to our motion and our interaction in the world around us? That's a subject that biomechanics is interested in. Enzyme networks are sort of related to gene regulation. Neural networks is a very broad term. Everything that has to do with the brain and how the brain functions falls under the category of neural network questions. Drug delivery. How can we make delivery of drugs, for example, cancer drugs, very targeted and very efficient? Cell fate. We don't know why cells do what they do. Cells in our body die every day. Cells in our body divide every day. Yet we don't know sometimes why they do it. Sometimes we do know. There's a lot of information, a lot of studies and experiments about why cells do what they do. But sometimes we don't know why they do it. Why a cell decides not to kill itself, not to terminate itself, but instead to decide to allow itself to become a cancer cell, for example. That's a question that people interested in cell fate study. And lastly, tissue tectonics. It's also a really interesting problem in biophysics. tissue tectonics deals with how we develop as embryos into how the cells, the stem cells from which we start, develop into the different body parts that we all have. Why did a cell decide to become my spine, and why did another cell decide to become my eye? That all is, the people who study tissue tectonics, these are the kind of questions that they try to answer. So I said something about protein dynamics a moment ago, and I want to show you an animation which I found on Wikipedia, which I think is quite illustrative. In front of you, you have a simulation. This is not a real protein. It's a simulation based on someone writing code and approximating how a real protein would work. And what you have is you have the different amino acids inside the protein that make up this long molecule. If you take this protein and stretch it, it's going to be pretty long. But the protein, which is just a long molecule, doesn't do anything. It has no effect, doesn't have any function. Long molecules are sticks. What do you do with them? Instead, proteins fold onto each other. They develop secondary and tertiary structures, which then give it a function. And the way they do it is pretty interesting. Let's see if I can activate this. And it's a question of study. People are really interested in how this is happening. Why do proteins fold the way they do? And what kind of function do they gain by getting these folded shapes? Because we know that the shape has a lot to do with how the protein functions and what it does. All right. Tissue morphogenesis, tissue tectonics. This is actually work I have done. This is an image that I have generated when I was a postdoc in Montreal. I know it's a little bit difficult to see, but this is a microscopic image of a slice of a live tissue, actually, but it's a confocal microscopy, meaning there is a virtual slice in an embryo of a quail, and what we can see are the cell nuclei. All these white dots, these numerous white dots, are all these cell nuclei. And what we did, we studied the motion of these cells, because these cells, as they move, they enter different regions of the future quail and start to differentiate in the different body parts. For example, what you see here is the spinal cord that is being formed right now. So cells are migrating to the left, and as they migrate, they decide that they are going to become a part of the spinal cord. Or over here, they're going to become a vertebra. They're going to harden up and become a bone. So all of these decisions and how these happen are studied by both physicists and biologists. I was a physicist working with biologists, analyzing this data. Biologists were taking the images, and I was creating the arrows. I was figuring out the motion of the cells within the embryo. Another interesting problem. Again, I'm just throwing a bunch of interesting problems that I at least find interesting in biophysics. I looked at what was out there and what might be interesting to different people, and finally, I just decided that these are the kind of things I want to talk about. And how many of you have seen this dress on the internet? It became a big meme a couple of years ago. Just one person? In the back, two people. All right, then let me ask you this. How many of you see this dress as white and gold? How many of you see it as black and blue? So we see it different. I actually see it as white and gold, and I have a really hard time believing that anyone else can see it as blue and gold, blue and black. But the truth is that this is exactly what it is. When this kind of image, and it's not the only image, I just didn't have enough space, but I could have added other images where you see this discrepancy. The same image is seen by different people differently. And the reason for that is actually still not completely understood. It has to do with how we perceive color. And it's an interesting question. A question maybe no one thought about for hundreds of years until fairly recently. How do we perceive color? Well, it turns out that our basic machinery in our eyes is very similar to how color TV cameras work. So this is how a color TV camera works. You have a lens, so the light is coming from the left, it hits the lens, and then, of course, the lens focuses the light, it creates crisp images, that's all good, optics, but then the light is split. Some of the light is run through a blue filter, and then recorded. So all the blue light is recorded in the TV camera. Some of the light goes through a green filter and gets recorded. And some of the light goes through a red filter and gets recorded. And then these three channels, RGB, red, green, and blue channels get recorded, sent over the air to your TV, and then your TV redisplays them back, recombines them back, and shows them on the screen. And that's how you see color. So it turns out in our own eyes, we actually have cones, these cone-shaped cells, which are sensitive to different parts of the spectrum. Some cones are sensitive to blue light, some cones are sensitive to green light, and some cones are sensitive to red light. Our eyes work very much like a TV camera. But then it gets more interesting. So if our eyes work all the same like a TV camera, why do we see that dress on the previous picture differently? And that's where it gets interesting, because the biologists tell us that there are three types of cones in our eyes, yet we know from a very simple experiment, I just took a poll in the room, that we actually perceive the color very differently, even though we all have the same machinery in our eyes. And it has to do, most likely has to do with how our brain processes that information, how the brain takes these signals coming from our eyes and then recombines them. In a TV camera, that's very simple. You have a bunch of digital electronics, a bunch of transistors, millions of them, in electronic circuits, and they recombine the signal, and every single TV camera and every single TV set does the same thing, with very few deviations one from the other. Some manufacturing errors. Otherwise, if you look at 2, 3, 5, 1,000 TVs, they're pretty much going to display almost the same picture. Yet we see things very different. And why is that? Still apparently not a completely solved question, even though there are some papers, I haven't read them, but there are some papers that have been coming out lately that sort of propose how this might be happening. Now, I'll talk at the end about, well, not at the end, in a few slides, I'll talk about why some experiments in biophysics are different and why is it so difficult to really get a clear answer to some of these problems. And it has to do with the fact that we're dealing with living organisms. You cannot just take a living brain and dissect it and see what happens inside. It's just not possible. All right. Oh, here are the slides that I want to talk about. I want to say something about the differences between biology and physics. The main differences, at least the differences that I see as being really obvious. So biology typically studies individual components of complex systems. We all agree that all living systems are very complex. And we contain a lot of components. And typically biologists study these individual components. They learn about the genes that activate these enzymes and those enzymes. We learn about the different receptors on the surface of a cell. Biologists study these individual components, but it's clear that these individual components work together as a group. Biology is a descriptive science. Biology observes things. Here is a machinery, here is a living cell. Biologists look at what's inside of it, they look at how it works, and they record it, and they observe it. And a lot of time and effort is spent by biologists figuring out smarter and better experimental techniques to be able to observe these living cells in their natural environment. Physics, on the other hand, is a predictive science. Physics observes phenomena, tries to explain them, typically with a bunch of equations, and then tries to make predictions about future experiments and future phenomena that we might observe. The ultimate goal of physics is to create a very compact set of equations, a very compact, synthesized understanding of the universe. The opposite of the quote that I started with. We don't want to build a machine, a computer bigger than the universe itself to try to explain the universe. We want to do the opposite. We want to write as fewer equations as possible in order to explain everything around us. And different subfields of physics then would arise from this grand unified theory as approximations of what's happening, of how the universe works. Not as exact description, but simply as an approximation. We have been somewhat successful with that. In front of you, oops, I didn't mean to do that. What we have here is what is called the Lagrangian of the standard model. Have you heard about quarks? How many of you have heard about quarks? All right, so quarks are the building blocks of the protons and neutrons inside the atoms, inside the nuclei of the atoms, which themselves are the building blocks of the molecules, which themselves are the building blocks of us. How quarks operate, how they interact with each other, how they glue to each other with gluons to become the protons and neutrons inside the nuclei. That all is explained by this very long expression, this very long equation. I think I've seen it. I think this is the first time actually I've seen it. I had to Google it and pull it off the internet to see it in all its full glory and how long it is. This is a physicist's ultimate dream. Being able to write something like this, even if it's bigger, longer than this equation, but something which will then be able to explain everything in the universe. Even if you go to the other end of the universe, you travel millions of light years away from Earth. If you want to still be able to arrive there, pull up your notebook with your equation, do your calculations, and then say, I expect this to be happening tomorrow on this planet, millions of light years away from the Earth. That's the ultimate goal of physics. Whether it's really an achievable goal, no one knows. But that's what we strive to do. And that's the difference between physics and other subjects like biology, because we do aim to get this very, very compact description of everything. We want to really understand everything in as simple a form as possible, if you can call this simple. As I said, this equation by itself, the equation I showed you, even if we can discover the ultimate equation, it's not really going to be that useful to us. Instead, we need to look at some approximations of this equation, make our lives easier, and start to look at ways of observing systems at different scales in a simpler manner. And this is where my work is really related to. I am a statistical physicist by training. And statistical physics is one of those approximation sciences. Statistical physics studies very complex systems with many, many components, like a living cell. And because we have so many numerous components, they don't have to be complicated, but there's many of them. It's difficult to study each component individually. Instead, we want to think of groups of components, what are called ensembles. And statistical physics is really good at explaining complex systems when you have these ensembles of components that are working together to achieve a goal. One of the examples that I like to give is the Meissner effect. What you have in front of you you have a piece of superconductor, something which is very cold, and a magnet. And what's happening is the superconductor pushes away the magnetic field through its bulk, and the result is levitation. This is something which has been actually quite well understood in physics. We understand that effect very well. How does a normal piece of metal work? A normal piece of metal contains atoms and conducting electrons, which are just floating around, moving around. See if I can pause this. Oops, sorry. See if I can pause this. And the electrons are moving around, but they bump into the atoms. Particularly, they bump into defects of the structure of the atoms. But whenever the temperature drops below a specific value with a specific magnetic field, external magnetic field, what will happen is that these electrons start to talk to each other. And they form what are called Cooper pairs, electrons that work together to overcome the resistance of the atoms around them. They work very much like the way you'd have a tow between two vehicles on the highway, right? I know some people have tried this. I don't recommend that, but if you can get really close behind a big semi on the highway, your actual mileage, MPG, increases because some of the air resistance of the air that would otherwise be hitting the windshield of your car gets removed by the truck in front of you. So it's a similar effect with Cooper pairs. Electrons work together to overcome the resistance of the material to which they're flowing. But in order to do that, the electrons have to communicate. They have to work together. They have to correlate and coordinate. This means that they work as a group. They have the same goal. They're trying to move through the crystal structure of a metal, and they do it together. They work together, they communicate through what are called correlations. All right, so statistical physics deals with very large systems where it's not possible to track every single component. Instead, the goal of statistical physics is to reduce these numerous components into smaller number of bigger groups of such components. These macro observables are called order parameters. And I'll give an example again with magnets. On the left, we have Iron Man, a human with a metal exoskeleton. In the middle, we have a Superman who is almost human. One of them is attracted to the magnet, the other one is not. And the reason for that is because in one of them, the individual components that make up the metal exoskeleton, they're all working together. They're all lining up together in the external magnetic field of magneto. While in the same components of Superman don't. They don't work together. They don't correlate with each other, and they don't coordinate. And of course, the result is that overall, in Iron Man's armor, we have this group of elements that are all working together, and they all form an overall magnetization inside the iron. While in the case of Superman, we have nothing. All the different components cancel each other. They don't work with the magnetic field. And of course, the result is that Iron Man is the one that's attracted to the magnet and not Superman. So it's this interaction between the components, the small microscopic components of a system, that give rise to these macroscopic observations, these macro effects. The second goal of statistical physics is to reproduce the cause and effect relations, what I was talking about, the correlations between the different components of a system, using an effective theory. We're not trying to reproduce perfectly everything, we're simply trying to explain it as best as we can. And an example I like to give is what happens when you type on a computer. How many of you have ever opened a computer to look inside of it and change a component? A few of you. I like to open my computers, I like to clean the dust off, I like to replace components, sometimes I burn things up. It's a hobby of mine. But most of us, including myself, don't know everything that happens inside the computer. But we do know that a computer has the computer is this big box in the middle. There's a keyboard connected to it, and there's a monitor. And if you type on the keyboard, things appear on the monitor. We know that there is a correlation between our keystrokes and what the display shows us. The goal of statistical physics is not to understand every individual path that the electrons that leave the keyboard have to take inside the computer. We don't need to know. As a statistical physicist, I don't care if there is a CPU, memory, hard drives, input-output ports inside this computer. I don't need to know that. All I need to know is when I type a key, something shows up on the screen. And I look for that simplest theory that can explain that. And sometimes that simplest theory could be just, there's a monkey inside the computer that pushes the corresponding switch whenever it sees you press a key. And then the letter appears on the screen. This is an effective theory. A monkey in a box is just as good of a theory as the actual explanation with a bunch of expensive electronics, as long as it reproduces the same correlations, that when the key is pressed on the keyboard, you get something on the screen. And that's what we're trying to achieve. We're trying to achieve these effective theories. So, what are the takeaway points from what I've been talking about? Well, if you're studying a complex system, including a living cell, you want to ask the right questions, you want to find the correct macro description, you want to find out those groups of components that work together. And you want to find a theory that explains the experimental observations, an effective theory that explains what you're seeing that you can turn around and use for future predictions. One of the things that the statistical physicists study is how information gets transferred between different components of both inorganic and living systems. I looked at some definitions actually of what information processing is, and I pulled this off the internet. So some definitions are interpreting incoming information stimulus to make a response suitable in the context of an objective problem or situation, processing of information, especially the handling of information by computers in accordance with strictly defined systems of procedure, the processing of information typically by computer or by an organism so as to yield new or more useful information. It's a lot of gibberish. Passing information is pretty straightforward. You know something, you tell it to someone else. You are the source, there is a gap between you and the recipient who then receives the information. Simple as that, but it's actually not that simple. So, three components. There is a source of information, there is the transmission of the information that happens, and there is, of course, someone who has to receive the information. But the information that is received is never more. It's always actually less than the information given. How many of you have played broken telephone? Where you have a line of people, start at one end, say something very quietly, whisper something quietly, and then at the end, the last person has to shout it. And it's something very different from what the first person started with. Information works the same way. Every time we pass information, some of it gets lost. Now, when we think about information, we typically think about Google and its server farms. This is a server farm. I'm not sure if it's Google's or someone else, but you have thousands upon thousands of computers on these racks. They're connected through a network, and they talk to each other and to the world. And of course, they lose some information. They have to have ways to compensate for those losses. But that's how we typically think of information transfer. But that's not the only way we can transfer information. We can transfer information just by making a sign and actually doing a punctuation error on this sign. There are two ways of reading this sign. Hunters, please use caution when hunting. Pedestrians could be using the walk trails. Or you can say, hunters, please use caution when hunting pedestrians. Right? It's one punctuation error. The words are there, the sign is there, it's one comma that's missing, but changes the meaning completely. Information is lost. We sometimes don't think about it, but information is lost. Information works the same way at the cellular level, at the molecular level of living organisms. Whenever our neurons fire, they talk to each other. They release neurotransmitters in synapses, in the gaps between the different neurons. That's information transfer. The neuron that releases the neurotransmitters does it because it has to. It's decided that now is the time to release neurotransmitters. That's a piece of information that the next neuron then receives. That's a transfer of information. Not as obvious as asking Google for the weather, but it's very similar in the structure. You have a source, transmission, and a receiver. Signal transduction doesn't happen just between neurons. Signal transduction, information transfer also happens at every cellular level, at every cell, at the membrane of a cell. Cells can sense their environment. They know what's around them. For example, a bacterium knows that there's food around it in that direction by detecting some of that food, by sensing the tasty chemicals that it then consumes. And it does it using receptors on its surface. Binds to those, the receptors bind to the chemicals, and then the cell knows to do something with that. That's information transfer. The cell detects that on one end of its surface, there is food, there's none on the other end, so it better go this way. Again, reiterating that, we have a signal, a source of information, and a receiver that receives that information. But how do we measure the amount of transmitted information? How do we know, what do we mean? What's little information, what's much? How can we quantify how much information is lost? We do this, and I promise this is the only equation I'm showing today, we do this with some mathematics. Mathematics is an integral part of physics. I tell this to my students, sometimes they don't believe me. But there is a mathematical way of describing information. We have a really good way of measuring that. We need to quantify it, and we actually do that. We do it using mutual information, which is based on Shannon's entropy. Not going to talk much about that. I'm just going to say that there is an equation that we can use. What happens when we have a signal, but we have multiple receptors, multiple receivers? Do we get better information? For example, today I'm talking to you. I am that source of information, and you're all receivers. Now, at the end of the lecture, you can all get together and you can all talk to each other, and you can kind of recombine everything that I've tried to say. Maybe you find out that I haven't said anything, or maybe some of you missed something, some of you caught that, some of you missed. You are the receivers. I'm the source, and information is being transferred with a loss. But it's an interesting question, right? Okay, would it be good to have multiple receivers? Is it better to have a big audience, or is it better to have a small audience if you're trying to convey information? Well, clearly, if you just go home and you don't talk to each other, you won't improve your understanding because you haven't filled up the gaps in your knowledge. But if you talk to each other, then things can get pretty interesting. You might be able to exchange additional information, stuff that you have missed. Maybe some of the information that got lost by one of the receivers was received by another one, and it can be passed back up. How much time do I have? Carrie, how much time do I have? Okay, 10 minutes to finish, or 10 minutes for questions?
10 minutes, and then we'll have a few minutes for questions.
Okay.
Yeah.
All right. So we divide these kind of communications as extrinsic, also called extrinsic interactions, and intrinsic among the receivers. What is the motivation between? I will get slightly technical here, but again, I promise no more equations. Let's say we have two receivers of information. And these receivers are listening to a speaker or are measuring the concentration of some chemical in the environment. And what I'm plotting on this axis is what the second receiver has measured, and on this axis I'm plotting what the first receiver has measured. And in this pink circle, what I'm showing is the error or the uncertainty in what they've measured. In other words, what they have received, here is where the actual signal is, but this is what the two receivers can potentially measure. Their goal is to establish exactly the signal, find out exactly where the signal is, but instead, I have this big pink circle, and their numbers, their measurements fall anywhere in this big circle. If the circle was bigger, their measurements would be completely pointless. How can I improve on that? I mean, does it help to have two receivers in that case? Both of them have pretty large range of potential measurements for the same signal. It doesn't look very practical. Is there any way I can improve this? Here, it gets even worse if I actually vary the signal. The signal can vary, and the circle where the possible measurements fall will change with the signal. And let's say I take one measurement. I don't know where the signal is right now. I've hidden it from you. Because if I only look from the point of view of the receivers, all I'm seeing is a point somewhere on this plot. And if I look at this point and ask myself, where did this point come from? Which circle did it come from? Can I use this information? Can I figure out which circle it came from so I can deduce where the signal was? I'll have a really hard time. This point could be from the pink circle, from the orange one, or from the red one. I can't tell you anything about the signal. My error is too large. But what if the two receivers start talking to each other? And one of them says, hey, when I measure high, you better measure low, and vice versa. When I do that, I change the shape of the error regions. By introducing these correlations, this cooperation between the receivers, I change the shape of what they see. So suddenly, the error ranges, even though they're still significant, the area of these ellipses is not small, even though that the error has not been reduced. My ability to figure out where my measurements come from, where the signal is, now suddenly becomes much better. there isn't much overlap between the ellipses. So if I get a point over here, I know it came from the orange ellipse. If I get a point over here, I know it came from the red ellipse. So there was a paper that came out in 2014, so 7 years ago, that if you make neurons, if neurons are talking to each other in what I call with negative correlations, meaning when one neuron fires, it suppresses all other neurons, there was a claim made that that would lead to better information transmission. These are the kind of questions that that I study, I and my collaborators study. How do neurons transfer information? What do they do to improve their ability to transfer as much information as possible to the next neuron? And so we looked at a very simple system. We looked at a cell which has 2 receptors on it. And those 2 receptors can measure the concentration of some food in the environment. And we looked at what it means for the information transferred through the membrane of the cell, depending on whether the 2 receptors are talking to each other or not. I'll show you how, this is actually just an animation of what the model we proposed. And then, so what we have is, here is the membrane, the cell membrane is over here, and we have the 2 receptors. Here is the food. Whenever the food binds to one of the receptors, it activates the receptor closes and transfers that information to the inside of the cell so the cell can make a decision. So this is just an animation. I'll show you the actual simulation in a moment. So see how this didn't activate, didn't spend enough time. Let's see if it's going to do it here. No, it didn't do it. Now it did it. It activated the receptor. Notice something, however. I showed you how the food just attached or got bound to the receptor briefly, and then detached and left. It didn't activate the receptor. That's an example of what really happens, and it's an example of another source of error. If you're trying to measure the environment around you, you want to be able to check every single piece of food that comes your way. You want to be able to count everything. But if you have some uncertainty with your activation of your receptors, you have a problem, you have error. We had an event where food bound to the receptor, but it didn't activate the receptor before being released back. That's a source of error, that's a problem. So it's something we need to overcome. And this is why we have issues with transferring information at the cellular level. These kind of errors cause problems, cause noise, and cause problems with how much information we can transfer. How can we improve that? Could it be better if the different receptors talk to each other? This is a schematic. I won't go over this. It's just a schematic of what I had earlier, but this is typically a schematic. This is a schematic actually we published in the paper. Here is an actual animation based on the simulation we did. This is actual simulation with real data, with real numbers. It's not data, but it's simulated data. So you can see that some of the food can hop between the 2 receptors, allowing a communication between the 2 receptors. And the thing is that whenever food is attached to one receptor, it cannot be attached to the other receptor, so it creates a negative correlation. The 2 receptors are oppositely correlated. And so we thought, okay, if this is the case, we must have better information transfer compared to when the receptors, there's no communication, there's no hopping between the 2 receptors. All right. But it turns out that was wrong. And actually, our result was to demonstrate that it's the blue line here, the high line, shows the amount of mutual information when there's no communication between the receptors versus when there is communication between the 2 receptors. So it turns out that communication between components of a complex system just by existing is not enough. It could be actually detrimental to the amount of information transfer. You have to do a lot more than that. You actually have to learn about how to improve your information. And it requires energy. It requires effort by the components of your system. And that's why it's not easy to study these systems. All right, I think I want to stop now. I don't think I have too many slides left. I think I might have some more slides, but I think this is a good stopping point. How do I proceed from here?
All right, so lots to take in, but do we have some questions? I'll use the microphone here so everyone can hear.
Okay, you said that every time there is a transmission from a cell to another cell, there will be a loss of information. It seems like in the body, if you've got pain and that pain signal is going through your body, you're losing signal all the way. Do drugs reduce that or increase the loss of information and that therefore reduce the pain? Is that happening?
Possibly, yes, possibly. You can have both effects, improving information and reducing information transfer using drugs by changing the dynamics of the biomolecules that are involved.
Interesting. Yeah.
Second question, are there people at UW-Whitewater that are involved in the same kind of thing as you are? Can you work together with someone else?
Not as far as I know. I don't think anyone else is interested in information transduction.
Other questions? Yeah, did you have a question? Okay.
Are you working with a particular organism, or is this all theoretical?
It's all theoretical. These are all models. There isn't a specific organism. Studying actual organisms in their full detailed description is really difficult computationally.
Any other questions? No? Okay, we're going to go from physics this week to climate change next week. So wrap your head around that shift, okay? But today, please join me in thanking our presenter today for this very interesting presentation. Thanks, everyone.